Welcome to the Society for Development Biology Postdoctoral Seminar Series. We are excited to highlight the work of our outstanding postdoctoral members. Today, Daniel McIntyre from New York University Medical Center and Katheryn Rothenberg from University of Toronto will share their research. Each speaker will give a 20-minute talk followed by 10 minutes of question and answer. Please enter your questions in the Zoom question and answer box. So Dan did his graduate training at the McKay Lab at Duke University, where he looked at pattern development in sea urchin. He is currently a postdoctoral fellow in Dr. Jeremy Nee's lab at New York University Medical Center, where he investigates how niche cells and stem cells interact to form their primordial gonad in C. elegans. So a fun fact about Dan is that he started farming oysters with his wife, and they are predicted to have a nice crop this fall. So if you'd like to take it, Dan.
Hey, thank you very much. That was a really great introduction. I study the germline in C. elegans, and in particular, I study the embryonic gonad in the worm. And you can see that pictured here in this image. And today I want to tell you a little bit about research I've been doing on how the basement membrane that surrounds the gonad primordium impacts the quiescence of the primordial germ cells. And we think that's a really interesting question, broadly speaking, because a lot of what we're learning about how the basement membrane impacts stem cell quiescence is probably going to be applicable to stem cell systems in mammals as well. So we'll dive right in. The entire germline in an adult worm, which I'm showing here in red, is derived from this very simple 4-cell structure that's found in an L1 larva. And if we look at this in detail, every single C. elegans has exactly 2 primordial germ cells, and you can see those here in red. And each of those interacts with a single somatic gonadal precursor cell, which I'm showing in yellow. And for my talk today, there's a couple of really important things that I want everybody to remember. First is that each worm has exactly 2 primordial germ cells when they're born. And those cells, they remain arrested in G2, and they don't begin to proliferate until the worm hatches and begins feeding. And that's really important because the worm doesn't want to build its germline until it's in an environmentally favorable or ecologically favorable environment. So this structure forms about halfway through embryonic development when the 2 somatic gonadal precursor cells migrate to the primordial germ cells and actually extend their membranes over the surfaces and completely enwrap the primordial germ cells. And you can see this process of wrapping in this movie, where the green somatic gonadal cells are extending their membrane over the surface of the primordial germ cells. Now, I'm not going to talk a lot about the cell biological mechanisms that are driving this wrapping today, but I did want to show this movie because it's really the starting point for the research that I'm going to talk about in terms of primordial germ cell quiescence. And the third thing that you need to remember is that the entire gonad is surrounded by a basement membrane. And here I'm highlighting that with the laminin M cherry, and you can see it makes a nice bubble around the primordial gonad. And we don't know everything about how this basement membrane is built or what its composition is, but certainly we know from a lot of groups' work that it contains a lot of laminin, collagen, nitrogen, and probably also other basement membrane components as well. And it's important to remember from my talk today that laminin is a heterotrimer. It has 3 subunits, an alpha, beta, and a gamma subunit. And worms have actually a very simple complement of laminin subunits, which makes them a lot easier to study than, say, mammals. They have 1 copy of the beta and gamma subunits. That's called LAM1 and LAM2. And so if you remove either LAM1 or LAM2, you remove all laminin from the embryo. They do have 2 copies of the laminin alpha gene. They're called epi1 and LAM3. And that means that worms can produce 2 types of laminin heterotrimers. And specifically, the gonadal basement membrane only contains epi1 laminin heterotrimers. So anytime we remove epi1, we can really specifically affect the gonad and leave a lot of the other base membranes sort of not normal, but relatively intact. Now, the basement membrane actually forms a little bit before the somatic gonad wraps around the primordial germ cells. And you can see the sort of dynamic process of the basement membrane forming in an embryo here. This worm has an epi1 tagged with M neon green. And I do want to stop for just a moment and acknowledge Dave Sherwood's group at Duke. They have made this marker and a lot of other markers that I've used in the study. And I really want to thank them for sharing those. But what you can see is that the 2 somatic gonadal precursor cells shown here in magenta are surrounded by laminin pretty much as soon as they begin to wrap around the primordial germ cells, okay? And the result of this process that's happening in this movie is that you end up with the structure that I showed on the first slide, where you have two somatic gonadal precursor cells, each of which wraps around the primordial germ cell, which sits in this pocket in the PG or in SGP membrane. And then that entire structure is surrounded by a basement membrane. So my story today, I think, really begins with what I'll call an obvious hypothesis, which led to a very unexpected observation. So my hypothesis that I'll start with was that the formation of this basement membrane was really important for promoting the wrapping by the somatic gonadal cells. And I don't think that I'm really stretching too far out there by suggesting that a basement membrane is important for maintaining a tissue architecture. And it turns out that indeed, in the case of the worm gonad, that is exactly what happens. As I told you just a moment ago, normally there are two somatic gonadal precursor cells. I'm showing them in yellow. They flank and wrap around the surfaces of the primordial germ cells. When you take away laminin, that structure is very perturbed. You see that the SGPs are almost always unwrapped from the primordial germ cells. And actually, this picture of a laminin-1 RNAi embryo is pretty good. Both SGPs are actually with the primordial germ cells. Oftentimes, they're completely separated. And this phenotype is highly penetrant, and it happens when you deplete any of the subunits of the epi-1 laminin trimers that are in the gonadal basement membrane. And as a nice control, you don't see this phenotype when we look at a LAM3 loss of function. We also have been able to figure out that the SGPs interact with the basement membrane through a receptor called dystroglycan. Dystroglycan is expressed on the outer surface of the somatic gonadal precursor cells. Here I'm showing you a dystroglycan MDN green, as well as a plasma membrane marker for the SGP. And you can also see that when we look at a loss of function allele of dystroglycan, we get the same unwrapped phenotype as when we take away epi-1 or laminin from the gonadal basement membrane. This then suggests a mechanism where the SGPs first recruit laminin to form the gonadal basement membrane. And they then adhere to or interact with that basement membrane via dystroglycan in order to maintain wrapping or to maintain the architecture of this tissue. So that was the obvious hypothesis, right? But in the course of these experiments, I made a really unexpected observation, and that was that often worms that were depleted for laminin had more than two primordial germ cells. And you can see that here. This is a LAM1 RNAi worm, which has 4 primordial germ cells. And as I told you in the beginning, normally worms are supposed to have exactly 2 primordial germ cells until they have begun feeding. These worms don't hatch, they cannot feed. In fact, they're embryonic arrested. So this is very unusual, okay? And it happens when you deplete any of the subunits that are part of the gonadal basement membrane. Again, so this purple fraction that I'm showing, about 30 to 40% of worms have extra primordial germ cells. And so what this suggests is that basement membrane around the gonad actually has 2 functions, right? In addition to maintaining wrapping, it is also critical somehow for maintaining the quiescence of the primordial germ cells. Now, as I say, the first function, we have a pretty good idea mechanistically that seems to involve a receptor. It's called dystroglycan, but we didn't really have a good idea of how the PGC quiescence could be impacted by the basement membrane. And so to tackle this question, I basically started to work backwards from what we know about how proliferation happens in the adult germline and try and extrapolate how that might be inappropriately triggered in the embryo. Now, in an adult C. elegans, the germline stem cells proliferate as a result of notch signaling. And the way this works is that you have a ligand called LAG2 in the somatic gonadal niche, okay? That signals to GLP-1, which is a notch receptor active in the germline stem cells. And that activates several direct transcriptional targets, which favor self-renewal at the expense of differentiation. And I wanted to test if this pathway was being inappropriately activated in the embryo when you deplete the basement membrane. And so I looked at 2 different temperature-sensitive alleles of GLP-1. And when I shifted either of those alleles to the restricted temperature, we saw that they very significantly suppressed proliferation in the absence of the basement membrane. I further investigated this by looking at direct transcriptional target of GLP-1, Sigil-1, and we wanted to know if that target was being activated or upregulated when the basement membrane was depleted. And so I used a Sigil-1 promoter-driven GFP that's localized to the nucleus. And what I saw was that this reporter was significantly upregulated when components of the gonadal basement membrane were depleted. This experiment also gave me a really nice way to formally test whether you needed the SGPs to activate notch signaling, because I could do a little bit of clever embryology. I can take a laser and I can kill the precursors of the somatic gonad before the gonad forms. And when I did that, I saw that this activation of Siga1 GFP was completely lost. So that really indicates that the signals resulting in GLP-1 pathway activation are originating from the somatic gonadal precursor cells. So this suggests sort of an intermediate model where normally the basement membrane is suppressing notch signaling, and that's making sure that the primordial germ cells remain quiescent. When you take away laminin, somehow the SGPs activate notch signaling. That drives proliferation of the primordial germ cells inappropriately. There's lots of questions we want to know here, right? But for the remainder of my talk, I'm really going to sort of focus on two of these. I want to talk a little bit about what ligands may be activating GLP-1. And then I also want to talk about, maybe more interestingly for me, what basement membrane receptors are the SGPs using to sort of interact with laminin and control the activity of that notch ligand. But before I talk about actually either one of those, I want to pause for just a second, because this is right about the time when COVID hit. And when I came back from COVID last summer, I changed the way I did these experiments a little bit. I'd always wondered if the sort of limited extent of proliferation of the PGCs that I saw in a laminin-depleted embryo was simply because laminin beta depleted embryos arrest and don't live long enough. So the change I made was, instead of generally speaking depleting LAM1, I depleted EPI1, right? So EPI1 is specific to the gonad. Other basement membranes can use LAM3, and so those worms are not embryonic lethal. They survive to be sterile adults. So then I could do a time course. And what I saw was that when you deplete EPI1 RNA by RNAi, there's a very significant increase in the penetrance of this phenotype between 24 hours and 48 hours. All right, so from now on, the change I made is that I'm looking at 2-day-old embryos using EPI1 RNAi. And so that's why the extent of the germ cell proliferation is going to be considerably greater from here on out in the talk. So the first question that I proposed in the last slide was what ligands might be activating GLP-1? And the logical choice for this is LAG2, which is the DSL ligand that activates GLP-1 in the adult germline. And I looked at a loss of function allele of LAG2. And in combination with EPI1 RNAi, you do see a statistically significant suppression of this proliferation. But the difference is small, right? Suggesting probably that another ligand is really the driver of this proliferation, and LAG2 is simply modifying that. And that was a little bit disappointing. Actually, I really thought it was going to be LAG2, but it does tell us something useful, right? And what it tells us comes from the fact that LAG2 is really the only ligand that is normally expressed in the somatic gonad in the late stages of embryogenesis. So this is single-cell RNA-seq data. It is not my data. It is data from Packer et al. 2019. And I just pulled out the data pertaining to the somatic gonadal cells, and specifically the DSL ligands. And it was very clear that this is really the only ligand with major expression. And so I think what this is telling us is that one of these other ligands is likely to be transcriptionally upregulated as a consequence of gonadal basement membrane depletion. And I haven't identified what that ligand is yet, but I do think that that does suggest a mechanism. So a model for where we stand with this at this point in the talk is that you have laminin in the basement membrane, which seems to likely activate some basement membrane receptor and inhibit the activity of a notch ligand. This model makes a lot of sense based on the data that we have so far. And it turns out it has vexed me. It vexed me for months. And I'll explain why. According to this model, you would expect that activation of the ligand to the receptor would initiate signal transduction. So loss of function of a receptor, like say integrin or dystroglycan, should mirror the phenotype of loss of laminin, right? It should cause extra primordial germ cells. But it turns out every receptor we've looked at failed to do that. And I'm going to show you here data just from PAT3, which is the sole beta integrin in C. elegans. No proliferation, same thing with dystroglycan. Did look at a couple of other receptors as well, but it just drove me nuts until I realized that the problem was that I had to rethink my model. Okay, maybe these receptors are functioning redundantly, or maybe they might enhance or suppress PGC proliferation. And to make a long story short, what I'd like to tell you is that in fact, loss of function of integrin very significantly suppresses proliferation in the absence of laminin. So here's the data that supports that. Normally, when you take away EPI1 by RNAi, about 3/4 of the worms have extraprimordial germ cells. That's what we expect. A similar fraction have extraprimordial germ cells when we look at a PAT3 null allele over a balancer. But when we look at the PAT3 null allele on its own, there's almost no proliferation of the germline when EPI1 is depleted by RNAi. And so this suggests, I think, a very different model than what I have just proposed. This suggests that, in fact, integrin is actually activating Notch signaling. And the function of laminin then is to actually block some integrin activating ligand and keep Notch signaling off. Now, one thing that I haven't provided any data for from this experiment is really whether or not PAT3 is actually active in the somatic gonadal cells themselves. Because it could be, of course, that you need PAT3 in some nearby tissue like the muscle or the gut, right? And it indirectly affects the gonad. To address that, I used a tissue-specific degradation. And the way I did that is with the ZF1 ZIF1 degradation system. And just briefly, I'll explain this. You take the ZF1 domain from a protein called Pi1. And what that domain does is through an adapter protein called ZIF1, it recruits an E3 ligase complex and causes the protein containing the ZF1 domain to be ubiquitinated and degraded. Steve Armenti showed several years ago that we could use this to degrade other proteins by simply moving the ZF1 domain to those proteins. I'll also draw your attention to Josh Abrams' paper that just came out in eLife. He used this system extensively to degrade components of the PAR complex in the excretory cell. So if you're interested in how this works, I would really recommend checking out Josh's paper. Simply, in my case, I added the ZF1 domain to the endogenous PAT3 locus, and I drove the ZIF1 adapter specifically in the SGPs with an SGP-specific promoter. And when I did that, I saw exactly the same thing, right? Normally, you have proliferation in the absence of EPI1. That's true in the PAT3 degron allele on its own. When you cross in the ZIF1 driver, specifically in the SGPs, you almost completely block proliferation of the germ cells. And so that tells us that you specifically need integrin in the somatic gonadal precursors to promote PGC proliferation. I'll end by just saying that this really suggests an unexpected model. This is not how I thought this was going to work. What we think is happening is that laminin in the basement membrane is actually keeping some other ligand from activating integrin. Okay, and I suspect that what's happening is that that is another basement membrane component that normally scaffolds on top of laminin. When you lose laminin, that ligand can activate integrin, which activates Notch signaling, which causes the germ cells to exit quiescence and to begin to proliferate. There are a lot of things that we'd still like to know about how this works, things we're working on right now, and I'll just highlight a couple of them that I think are most interesting. First, we want to know what co-receptors and signal transduction mechanisms are operating inside the SGPs downstream of integrin. And that could be things like integrin-like kinase and tailin, for example, so that we're looking at those now. Also, we'd like to know what components of the basement membrane are actually interacting with integrin to sort of trigger quiescence. And preliminarily, I have some data that suggests that is collagen, but I'm not ready to say that formally yet. And then lastly, I think a really exciting aspect of this project is I'd like to know if remodeling of the gonadal basement membrane actually enables proliferation in later larval stages. Obviously, the worms, they need to have a germline, right? Once they find food, they need to have germline stem cells proliferate. And so I think that remodeling of the basement membrane may facilitate that process. And that I think is a really exciting question, for example, not just in worms, but also in cancer biology, right? Where changes to the interaction with the extracellular matrix can be key to driving tumor progression, tumor proliferation. I will stop there. Thank you all so much for your attention. I would like to specifically thank a couple of people, obviously Jeremy Nance and everyone in our lab. I would also like to acknowledge Jane Hubbard, who has taught me everything about the worm germline. I'd like to acknowledge Dave Sherwood's group for sharing reagents with us and say that we're funded by the American Cancer Society, NISTEM, and by the NIH. So yeah, oh, and this is the dock where the oysters are being grown, if anybody's interested. So thank you very much, and I look forward to answering questions.
Well, that was thrilling, and there was a lot of curves in that presentation. Must have been really fun. So I'll start out. So earlier in the talk, you talked about how there was 2 versions of the laminin, and if you knock out the EPI1, it only affects germline. So yeah. What's unique about the EPI1 protein that would make it germline specific compared to its homologs?
Right. Okay. So let me pull up the relevant slide here. Yeah. Great question. There are specific protein domains at the end of laminin alpha that are different between epi1 and lam3. And I don't know this in worms, but based on stuff in other systems, I think it's likely that differences in that protein domain affect how that protein gets recruited by cell surface receptors. So, for example, Dave Sherwood's group has shown that you need a particular integrin to recruit epi1 laminin trimers around the gonadal-based membrane. So I think that that's where the answer lies, and that's something that we're interested in looking at, but I don't have any data on that specifically.
Awesome. So we have a couple questions. So Yana Bokrishn, I'm not going to have that last name, but I'm sorry I butchered your name, says, great talk. Have you looked at BMP signaling in the PGC SGP in absence of a laminin?
No, I have not looked at BMP signaling in the PGCs or SGPs in the absence of laminin, but there's I don't have any evidence that that type of signaling is working at this stage normally. So I wouldn't necessarily expect laminin to affect that signaling. It does happen. It does have an effect later, but I don't think that that's happening now.
Michael Bradbury says, in the basement membrane remodeling, is epi1 lost or decreased as the gonad matures?
That is a really good question, right? And I don't think we have enough information about that to answer one way or the other. It is still there, but I can't really say if it changes in sort of the quantitatively, right? Like I don't have any information on that right now. I think that in terms of what changes in the basement membrane as the worm gets older, I suspect that the changes are going to be in some other proteins, but I don't know that, right? And there's some proteins that change, like different isoforms come up. So I guess I would say that we just don't know enough about that to really say if laminin is really like the key change as the worm gets older.
Peng Hong says, very nice talk. Maybe I missed it, but how does integrin activate the notch signaling?
I would really like to know the answer to that question. So one hypothesis that I'm testing right now is that there could be a transcriptional activation of a notch ligand. Okay, that's one model. I think the fact that it's not LAG2 sort of rules out some things that we had been considering, right? So we knew LAG2 was expressed, transcriptional reporters show it's there, sequencing data show it's there. So if it were LAG2, you could see it being regulated post-transcriptionally, you could see it be protein localization. But given that LAG2 seems to not be the main driver, like it seems most likely to me that one of the other ligands has to be turned down transcriptionally. So what you have is some sort of a signaling cascade where integrin, maybe through integrin like kinase or through tailin somehow activates transcription of a ligand in the nucleus. But that's really, a hypothesis at this point.
Anne says, Corsi says, really interesting work. When you said you had extra PGCs, they were often spread out. Do you think they are actually migrating like metastatic cells, or is it more passive in that the basal membrane is not keeping them anchored?
Yeah, the primordial germ cells are not actively moving like a cancer cell. Any sort of separation or rearrangement is being caused by sort of movements of other cells.
Oh, that's interesting.
And we have movies where you can see how these cells divide and get sort of pulled as the tissue gets sort of screwed up. So like that I can say with pretty certainty that they're not actively sort of moving themselves.
Because in the zebrafish, they migrate a long way themselves. So that's interesting.
Right. So this is the opposite of a zebrafish, right? Almost all the other animals that people think of, the PGCs migrate, but it is the SGPs that migrate to the germ cells in the worm. So it's the opposite of what you see in a fly or a zebrafish.
Ian Ching Seng says, nice talk. Did you test downstream signaling of GFP1 like LAG1 and LAG3 suppresses divisions?
Right. So did I test some of the other known factors that are downstream of GLP-1? No, I didn't. And the reason is just that I've been much more interested in how these cells are interacting with the basement membrane. So like I didn't sort of, absolutely exclusively or, without exception, test every downstream component. Because for me, like, my interest lies more in sort of the cell biology of how these things are working with the basement membrane and in sort of the factors that are actually driving proliferation.
So that's all the questions we have, but I have a couple more. So
Good.
How are you going to figure out what is the ligand that's responsible? It seems like there's a lot of choices there. Are you going to like knock them out systematically? Are you going to do like a sequencing approach, like an RT approach, or do you have like a hypothesis of maybe there's a couple?
Yeah, so the initial hypothesis is to look at, there's a couple of other ligands that play minor roles in driving adult germline stem cell proliferation. So we're getting transcriptional reporters for those, and so we're gonna look at those first. However, we can also get at this with a sequencing-based strategy. So one thing we're gonna be doing is just sequencing the somatic gonadal precursor cells from worms where the base membrane's been depleted, right? And if there's a transcriptional upregulation of the ligand, we should find it that way. We may also find a lot of other interesting things about how these cells are interacting with the basement membrane.
Do you think it could be that there's mini ligands that could either contribute or like compensate for this in this situation? And that's why you're only seeing a partial knockdown?
I'm sorry, can you ask that one more time? I missed it.
You only see a partial knockdown when you knock out the main one. Do you think that the other ligands are contributing to it, or are they compensating?
I think that I think that another ligand is likely to be the main driver, and LAG2 is contributing, right? Okay. So LAG2 is sort of adjusting the extent of proliferation, but the reason you get proliferation or not proliferation is because some other ligand is being activated.
That makes sense, for what I know. Do you have sorry, I was just trying to think of the best way to put this. Do you think that the extra primordial germ cells are cells that are around the PGC adopting the PGC cell fate, or do you think it's that the PGC cells are dividing?
The PGC cells are definitely dividing. You can see that in movies.
Okay.
Yeah, so there's some more data on how those cells actually divide that I didn't show, just trying to make time for more recent data. But yeah, we have movies that you can watch them divide.
Can I ask a question while we have a minute left?
Yeah, Brendan.
When you laser ablate the PGCs, what happens to the somatic gonad? Does it fall apart? Do distal tip cells still migrate? Just curious.
So my experience has always been laser ablating the somatic gonadal cells. And when you ablate the somatic gonadal cells, the primordial germ cells do one of two things, right? So either those cells can actually die, and there's this crazy mechanism. I didn't talk about this here because it's talked about it elsewhere. But what happens basically, normally the primordial germ cells in a worm make these lobes, and the lobes are actually engulfed and degraded by the endoderm. And so what you see here is this is sort of a diagram of the actual structure in a worm embryo. You have the PGC cell body, it's surrounded by an SGP, and it produces this lobe that is eaten by the endodermal cell. If you laser ablate the somatic gonadal cells, the endoderm can actually eat the primordial germ cell body instead of the lobe, killing the primordial germ cell. So that's a really weird thing that we figured out when we did this. It doesn't happen in every embryo. And in fact, if the PGC isn't eaten and you've laser ablated the somatic gonad, the PGCs actually, they just hang out, right? Like they're just kind of like, they're just kind of there. A great question is I've never actually grown one of those worms out to be like an adult to try and sort of like track the primordial germ cell, but that might be a really cool, interesting thing to do.
We have one more question, and it's from Bob Goldstein. Can you predict which PGCs will divide based on their positions near other cells?
Ooh, I haven't seen any bias in terms of whether the, for example, more anterior or posterior PGC divides first. But I don't think that I've really looked carefully enough to know. I think the problem there is just that, like, it's really hard to get good movies of a worm at the stage where this is going to happen, right? The worm is in the eggshell moving around like a sort of like swimming pretzel in the eggshell. And that makes it incredibly hard to get like reliable imaging of it, right? So that's a problem. And by the time the worm hatches, when you can paralyze it and then get a really good image, like the one I was showing in that last slide, then it's too late, right? They've already started to divide. So I think that I'm hampered a little bit just because like we really need to be able to image them, at the pretzel stage, and that's devilishly difficult. You need a really, really good lattice light sheet, and even then it's not perfect. But it's a good question, right? Like, yeah.
Well, thank you for that wonderful talk, and good luck with your oysters.
Thank you so much. Let me stop sharing my screen.
All right. Thanks, Dan. So next up, we have Katheryn Rothenberg, who is currently a postdoctoral fellow in the Fernandez-Gonzalez Lab at the University of Toronto. Katie obtained her PhD in biomedical engineering from Duke University, where she studied the role of vinculin load and dynamics in mediating cell sensing of physical cues. She did this in Brenton Hoffman's lab. Her doctoral work was supported by a graduate research fellowship from the NSF. She continued this scholarship at a high level as a postdoctoral fellow. Katie's been awarded the Canadian Institute for Health Research Fellowship in support for her research, which investigates mechanisms underlying collective cell migration and cell extracellular matrix adhesion in embryonic wound healing, which we're going to hear about today. So without further ado, I will let Katie take it away and tell you about her exciting findings.
Great. Thanks so much for that introduction, Brandon. Thanks for the opportunity to talk to you about my work. So in the Fernandez-Gonzalez lab, we're very interested in collective cell movements and how cells are able to work together to move and form tissues and things like that. And so obviously there are plenty of examples of really elegant collective cell movements during embryonic development. For example, in zebrafish apibole, this enveloping layer migrates as a collective to enclose the yolk. Also during Xenopus gastrulation, you have the involution of these marginal zone cells that move through the interior of the embryo to create the multiple germ layers. And what's really neat is that these processes share some similar features, including kind of their maintenance of contacts with each other, their neighboring cells, and with the environment, as well as use of the actomyosin cytoskeleton to generate contractile forces that help to drive migration. And these are also shared with many migratory events during disease states as well. So we think understanding these cell adhesions and cytoskeletal rearrangements are really important for understanding how any of these processes happen. And so the model that we use in the lab is the Drosophila embryonic epidermis. So here we have a section of the epidermis at stage 14, which is about halfway through development. And here we have the cells outlined in magenta, which is marking E-cadherin, a component of the adherence junctions. And then in green, we have the non-muscle myosin 2. And so we generate wounds in the epidermis using a high-power UV laser to ablate 3 to 4 cells in the tissue. And what that does is then that creates this wound, this gap in the tissue that the cells then organize and move forward into the gap to close it. It's a pretty rapid process. It occurs, wounds are healed within 45 minutes to an hour. There's no cell divisions, so it's fully these cells moving in to close the space. And then at the end of the process, there's no scarring. So yeah, we find it a really nice model system for studying collective cell migration. And what you'll notice in this movie is that there are definitely rearrangements of the E-cadherin and the myosin as the cells migrate. And most notably, we see accumulation of myosin at this leading edge. And so together with actin, this forms this contractile actin-myosin cable that helps to both kind of coordinate neighboring cells as well as drive their motion into the wound area. And so what's not as visible on the movie, but what we can see when we look at individual images is there's also this striking rearrangement of adherence junctions. And so previous work in our lab has shown that E-cadherin and other adherence junction components are endocytosed from the leading edge. And so you get a depletion at these, what I call linear junctions at the leading edge, and you get accumulation and reinforcement of former tricellular junctions. So here marked with these arrows, you see junctions that used to be between 3 cells. That third cell became part of the wound, and then that remaining junction is reinforced. And what's interesting is there's definitely crosstalk between the rearrangements of the adhesions and the cytoskeleton. For example, if we block endocytosis of E-cadherin using Dinosaur, we see that there's a defect in the ability of the wounds to close, and we don't actually get formation of the actin-myosin cable here that's shown in red. And so there's definitely interplay between these rearrangements. And so just as a visual summary of what we know so far, is that adherence junctions are removed from these linear junctions at the leading edge and accumulate at these former tricellular junctions. And then simultaneously, actin and myosin are assembling a cable that will encircle the wound edge and help to drive contraction. And what we noticed was actually interesting about this cable is that it's heterogeneous in the early stages of wound closure. So you'll have regions where there's higher accumulation of actin and myosin than in other regions. And through some recent work, we've shown that this heterogeneity is actually required for wounds to close rapidly. So having patches of myosin-rich and myosin-poor, actomyosin-rich and actomyosin-poor areas around the wound edge is actually important. And in mutants that we lose this heterogeneity, we have defects in wound closure. And so how this kind of contributes to the ability of the cells to close the wound is through a couple of different mechanisms. We found through other work that, regions with high actomyosin localization are very contractile and under high tension, and that actually stabilizes actin and myosin at those junctions. So areas that are already under tension are stabilized and will maintain high accumulation. And then those contracting regions can strain their neighboring actomyosin poor regions. And we found that strain actually induces recruitment of actin-myosin. So having this heterogeneity enables this kind of ratcheting mechanism to continually polarize actin-myosin to that leading edge. And so at this point, we know kind of a decent amount about what rearrangements are occurring in cell adhesions in the cytoskeleton, the timing of these rearrangements, and how those contribute to wound closure, but we don't really know what's driving the rearrangements themselves. And so I became interested in the small GTPase RAP1 because it's known to be responsive to mechanical stresses, which we definitely have at the wound edge, as well as reactive oxygen species, which other work from our lab has shown is important in driving the wound closure response. In turn, RAP1 is known to interact with cell-cell adhesions, cell matrix adhesions, and the actomyosin cytoskeleton in a variety of systems and through different effector pathways. And so to me, it seemed like a good candidate for kind of mediating these simultaneous rearrangements that are really driving the forward migration of these cells. And so in this talk, I'm going to tell you a bit about what we've learned so far about how RAP1 is mediating or interacting with cell-cell adhesions and the actomyosin cytoskeleton. And so a brief overview of the genetic tools that I'm using. I have a RAP1 dominant negative construct that will reduce RAP1 activity. as well as a constitutively active construct, which will increase RAP1 activity. And I use the UAS SCAL4 system to drive these in mid to late stage embryos so that they're turning on a little bit later and should be highly active during stage 14, which is when we're imaging. And so the first thing we looked at was just whether RAP1 activity has an effect on the rates of wound closure. And so here I'm showing the same markers again, magenta showing E-cadherin and green is myosin. In our control, we see what we expect, a nice accumulation of myosin at the wound edge, or a rapid closure within 45 to 50 minutes, the wound is closed. And we don't see this with the dominant negative. We see a much slower wound closure rate. So at each time point, the wound is larger in the RAP1 dominant negative. And we quantify this by first looking at the mean area of the wound over time. And what you'll see is that upon wounding, because there's tension in the tissue, there's an initial recoil. And then as the cells reorganize and start moving forward, there's a fast phase of wound closure and then a slow phase. And what you notice in the dominant negative is that, first of all, there's a little bit of a delay in the initiation of the closure, and then it occurs much more slowly. And so we quantify the rate of wound closure by fitting individual curves with an exponential decay function and extracting the time constant. And when we compare those between these two groups, we see a significant decrease in the rate of wound closure when RAP1 activity is interrupted. In contrast, when we overexpress the constitutively active RAP1, we increase the rate of wound closure. So we see these wounds closing in about 30 to 35 minutes. And when we quantify the closure rate constant, we do see a significant difference. And so we rarely see things that make wound closure happen more quickly, since it's already a pretty rapid process, but this does. So it suggests to us that RAP1 activity is, activity levels are pretty important for mediating this wound closure response. So then, now that we've confirmed that RAP1 is playing a role, we want to delve more into what that role might be. So the first thing we looked at was, where is RAP1 localizing during this process? So as I mentioned before, we know that E-cadherin localizes to these puncta around the wound that are sites of former tricellular junctions. And when we quantify the intensity at these adhesions over time, we see an increase of about 20% in E-cadherin intensity at those sites. And when we look in the same embryos and the same wounds at RAP1 intensity at those same puncta, we see a similar increase in intensity. So we again see about a 20% increase at those junctions. And so to get an idea of whether these components are arriving at the same time, or one is preceding the other, we did a cross-correlation analysis between the two traces. And we found that actually the maximum correlation occurred at a time shift of 0. And what that means is that within our time resolution, which here is 15 seconds, RAP1 and E-cadherin seem to be arriving at these tricellular junctions at the same time. And so thinking about whether these could be moving in a complex, we started thinking about a RAP1 effector called canoe in flies. It's known as aphodin in mammals. And this is a known RAP1 effector, and then it also is known to interact with adherence junction components. So we looked at canoe localization during wound closure and saw something very similar. Again, we have E-cadherin accumulation at these former tricellular junctions. We also have canoe accumulation, again, about 20% increase, and they do seem to be arriving at the same time based on our cross-correlation analysis. So this kind of led us to believe that RAP1 may be acting through canoe to position adherence junctions at these former tricellular junctions. And so to get at testing that, we looked at the effect of RAP1 on cell-cell adhesion localization. And when we express the dominant negative RAP1, first of all, you'll see overall there's a decrease in E-cadherin at the cell-cell junctions, and that is expected based on the literature. RAP1 is important for formation and maintenance of adherence junctions, so it does make sense that we'd see an overall defect. But if we compare relative to these initial levels, and we look at linear junctions, so along the wound edge, and tricellular junctions, we see different effects. So if we look at these linear junctions, in both cases, we see a loss. And so that suggests that RAP1 is not responsible for the endocytosis of EK adherent from the leading edge. However, at the tricellular junctions, in our controls where we usually see these puncta emerge, in our RAP1 dominant negative embryos, we actually see a loss of intensity at those junctions. And so RAP1 seems important for positioning adherence junctions at these points and reinforcing them over time. When we look at the constitutively active RAP1, so increased RAP1 activity, we don't see any major effect on positioning of the cell-cell adhesions. So we thought that maybe there's more to the story than just cell-cell adhesions, since these are closing more quickly, regardless of similar EK adherent localization. So we began looking at how RAP1 may be interacting with the actomyosin cytoskeleton. And so first looking at a marker of F-actin, in our controls, we see this nice continuous accumulation of F-actin to the wound edge. You'll also see some protrusive activity. And in the dominant negatives, we see a defect. So we do see some localization to the wound edge, but not a continued accumulation that we see in the controls. And if we look at the percent increase from pre-wounding to 45 minutes following wound closure, there's a significant defect. And so that does seem to indicate that RAP1 is responsible for getting F-actin to the wound edge. And what's also interesting is that there are also effects on the heterogeneity. And so I described in the beginning how heterogeneity contributes to rapid wound closure. And so to kind of visualize heterogeneity, I made these chymographs, which are just linearized wound edges. So just kind of unraveling it, and then showing the intensity over the first 15 minutes of wound closure. And what we can see in the controls is that at the onset of wounding, we get these patches of high actomyosin accumulation. And so we see a spike in the heterogeneity. But over time, as these patches are contracting and straining their neighbors, we're getting additional recruitment of actomyosin to those neighboring areas. And we see overall a decrease in heterogeneity as a robust cable is formed. However, in the dominant negative, the picture looks very different. We do get the emergence of some patches of higher actomyosin accumulation, but over time, these don't lead to additional recruitment of actomyosin. And so we just kind of get a flat heterogeneity over time. So this seems to be some sort of defect in either forced generation or forced transmission that's not contributing to further polarization of actomyosin. And so to kind of confirm these suspicions, we looked at myosin accumulation as well. And so here again, with the RAP1 dominant negative, we see a defect in overall myosin accumulation. And again, we see some accumulation in patches, but we don't see further reinforcement. And so this does lead us to believe that RAP1 has some role in reinforcing the actomyosin cytoskeleton. If we look at overactivity of RAP1, we don't see a significant difference in accumulation from pre-wounding to 45 minutes post-wounding. But what we do see is that in earlier time points, myosin accumulation is higher in the RAP1 constitutively active. And so it seems that myosin is getting to the wound edges more quickly in the RAP1 constitutively active. So that does suggest that RAP1 may be responsible for this recruitment. And so having the actomyosin cable is important, and its ability to generate forces is also important. So we've been testing the tension in the cable around the wound edge, and we do that using single laser ablations. So we use our laser to cut one single linear junction at the wound edge and watch how fast the ends recoil. And so the more tension that segment is under, the faster it will recoil. And these are preliminary results, so still kind of bulking up the numbers here. But so far, it's looking like in the RAP1 dominant negatives, we have less tension at the wound edge, so we're not able to generate as much contractile force. And in the constitutively active, we have more. And so these are all at the same time point within wounding. And so at 10 minutes post-wounding, we have this variation in contractile abilities of the actomyosin cable, which may set the stage for different rates of wound closure. And so a story that I hope I've told you today is that we think that RAP1 is responsible for the reinforcement of cell-cell adhesion and actomyosin structures. Without RAP1 activity, we still do get endocytosis of E-cadherin, we get some polarization of actomyosin, but we don't get that further reinforcement of these cell-cell junctions or a robust actomyosin cable, and as a consequence, slower wound closure. And so we feel relatively confident that RAP1 is interacting with adherence junctions through canoe based on our results and based on the literature. We're working on experiments to confirm that. But what we're not sure is whether RAP1 is directly affecting the actomyosin cytoskeleton or whether the reinforcement of these junctions is what enables the actomyosin cytoskeleton to form a nice cable. And so our next steps will be trying to differentiate between these two prospects. And so with that, thank you for your attention. And I'd like to thank my advisor, Rodrigo, and the rest of my lab for creating a very supportive environment, as well as our collaborators who've provided us reagents and ideas, and of course our FLY resources and our funding sources. And I'd be happy to take any questions.
All right, great talk, Katie. I'll guess I will start it off. I'm really interested. You did the wound, you did the laser ablation in mid to late stage embryos in the epidermis. How tissue specific can you be with those wounds? Can you wound other tissues and see similar effects? And do you think it could be mediated by RAP1 in the embryo? And then in adults, can you also go into the epidermis and injure? I'm sure you probably get this question all the time. And do you see wound healing at all in any adult tissues? Just curious.
Yeah, that's a good question. I don't know about adult tissues. We have wounded the epidermis at other stages of embryonic development, and the mechanisms are actually fairly different. I'm not sure about RAP1's activity, but in early stages of development, there's more involvement of the cells actually in the wound, contributing to the wound closure. And then I know there is some work in larval stages of wound closure. I don't think anyone's looked specifically at RAP1, but I believe that those wounds leave scars. And so they're not, there are some different mechanisms, especially in the resolution of the wound at the end. But yeah, we haven't personally looked at RAP1 in those systems, though other people have looked at RAP1 in other collective migratory processes and seen similar results.
Okay, perfect. So Joshua Abrams says, nice talk. Do you have any ideas of how RAP1 is activated at the wound edge?
Yeah, I think we have a couple different ideas. I think at this point we focus mostly on what's downstream of RAP1, but we looked at one RAP GEF called Dizzy or PDZ GEF, and I knocked that down and didn't see any changes to wound closure. But there are other GEFs and gaps. So in particular, I'm interested in EPAC, which is a GEF that actually could be responsive to reactive oxygen species. As I mentioned, those are definitely present during wounding and can induce some of these same rearrangements that we see. It's also possible there could be sensing of, changes in tension at the wound edge. And so I'd be really interested to see, there's, at least in mammalian systems, I think it's called C3G is a GEF that's known to be sensitive to the mechanical stresses. And so I think any of those are possibilities, and I definitely look forward to exploring them.
Right. So Raul Ramos says, great talk. Has anyone explored this RAP1 actin axis in scarless, rapidly healing vertebrates such as axolotl? How about mammals?
I'm not familiar of any studies of RAP1 in wound healing in those systems. Yeah, I know of other cell migration systems. Most of them have been in culture. So yeah, I'm not sure. I think it could be really interesting to see, since in those systems there are a lot more studies on RAP1 interacting with cell ECM adhesions, which we haven't really looked at in our system. And cell ECM adhesions may be playing a role in scarring and things like that. So I think that's definitely an avenue that could be pursued. I'm not familiar with any work so far, though.
Okay. So Michelle Stars Guiano says, nice work. Do you know if the molecular heterogeneity around the wound comes from different starting positions for the cells involved?
Yeah, that's a good question. So I have some theories about it that we haven't fully investigated. Let me see if I can find a good example of this. So there are kind of these cables running through the epidermis at this stage. And so actin and myosin are planar polarized in these stages. So while we haven't looked too closely at this, it could be that the initial accumulations are coming from segments of cells that were part of these cables. And so they're kind of already have actin-myosin polarized. So that's kind of one idea. It could also be dependent on different mechanical forces that the cells are experiencing at the very onset of wounding. If they're stretched more or less than their neighbors, maybe they immediately respond. But yeah, I think that's a great question and something we should delve into.
Okay. Cara, the other moderator, asked, do you think this mechanism would apply to tissues that have been damaged in other ways? For example, like a burn?
Yeah, so we've done a bit of work to show that these, at least the cytoskeletal adhesion rearrangements are present when we do like poke wounds with a needle. And yeah, they do look similar. So I imagine most forms of tissue damage are going to create some of these same signals that are going to drive cell migration. So I would imagine it should be a pretty conserved process. All
right. And that's about perfect on timing. So I want to go ahead and just close things out. And I want to say thank you to Dan and Katie for your excellent talks. The seminar has been recorded, just as a reminder, and will be available on the SDB website starting Monday. Please join us for next month's seminar. It'll be Friday, June 11th, when Elise Jarvella from the University of Maryland and Ryan O'Neill from the NIH will present their research. Thank you all for coming. Thank you for the great questions, and have a great weekend.